So, finishing up our discussion of the innate immune system, you're probably familiar with the concept of inflammation and some of the cardinal signs: swelling, heat, etc. But you might not know what are the causes specifically, and what is the purpose of inflammation. So inflammation occurs when there's a challenge from bacteria or a virus, as well as just generally trauma or toxic substances. So inflammation can be a response to those, and the purpose is to wall off the affected area. So whether this is damage, something like a burn or a cut, or whether there's actually an infection going on, that's going to trigger the release of inflammatory mediators. And that inflammation is going to serve to wall off the damaged area. So it becomes a containment issue. In addition to containment, there's also cleanup that has to occur. So what's going to be doing this cleanup is a combination, a combined effort by the cells and the proteins that are being released, that some of which we've already discussed, phagocytes and other proteins related to an immune response are going to be there to clean up the affected area. So inflammation, again, is a response to some tissue damage or a challenge from some pathogen. So let's envision a scenario where you have tissue X. So I've drawn kind of a layer of cells here. And in tissues, you're also going to have cells, some monocytes that are those phagocytes that we mentioned, and also cells like mast cells that are involved in the innate immune system. But they're already going to be present in many tissues so that they can respond quickly if there is any trauma or a challenge from a pathogen. So let's imagine that you have some trauma, let's say a burn, for example. So you burn this tissue, and that's going to do damage to these cells and cause them to release their own inflammatory mediators. So those cells kind of are ruptured and release these inflammatory mediators that serve as a signal to these local mast cells. That term local means these are cells that are just typically found hanging out in random tissues. And they're there kind of as like patrolling police officers would be there in a case of an emergency. So these mast cells are going to receive that signal and release their own inflammatory mediators. And so this is how the alarm is raised if there's damage to a tissue or introduction of toxins or pathogens. And these inflammatory mediators, what they do is they mediate inflammation primarily by acting on blood vessels. So if you think about the cardinal symptoms of inflammation being redness, heat, swelling, and pain. These are going to be related to the release of inflammatory mediators. So let's talk about the pain first. Inflammation is painful, and that helps to alert you to the presence of damaged tissue. It's good to know if you have a challenge that a certain part of your body is dealing with. And so we're alerted to the fact by the presence of pain. So if you look at some certain inflammatory mediators, bradykinins and prostaglandins, are going to cause sensory neurons in these areas to become more active. So sensory neurons transmit the feeling of pain to our central nervous system. And so those becoming more active these are actually going to make it easier for those neurons to depolarize and send signals of pain. That alerts us to the presence of an issue in the tissue in question. So that deals with pain, but these others, swelling, heat, and redness, are going to be directly a result of what's happening to these blood vessels. So we mentioned one of the, swelling is one of the features of inflammation, and that helps to wall off a damaged area. And that swelling is brought about because of increased blood flow. So you have vasodilation. Of arterioles, and that's going to promote, increased blood flow through the capillaries that serve this tissue. So that vasodilation and increased blood flow is going to cause an increase in the swelling. The more blood you have flowing, the more loss of volume of the blood is going to happen at the capillaries. We've already talked about pain. Blood is also going to be a higher temperature than the surface of your body. And so the more blood flow you have, the more heat is going to be present in that area. So the heat is also going to be increased with that increased blood flow. And then redness. The more blood you have in an area, the redder it's clearly going to be. So that would be the reasons for seeing these cardinal signs of inflammation. As far as the swelling goes, histamine and bradykinin are going to be potent vasodilators. So the arterioles dilate, increase blood flow. And you're also, so with that increased blood flow is going to come increased leakiness of these capillaries. So normally capillaries, and they have, different varying degrees of leakiness depending on the tissue. But in general, capillaries are going to become more leaky. And this is because those repair proteins and cells that we mentioned need to be able to more efficiently squeeze out of these capillaries from the circulation. So increasing the leakiness as well as increasing the amount of blood flow is just going to get these first responders there quicker. So that's going to include phagocytes like your monocytes and your neutrophils. And it's also going to activate the proteins of the immune system, repair proteins and those complement proteins that we mentioned previously. So complement proteins are going to be activated and also be part of this initial immune response. So again, the four cardinal signs of inflammation are directly related to blood flow that's brought on by the release of inflammatory mediators. And these can also have an effect on transmission of pain signals. An additional effect of inflammation and the presence of these inflammatory mediators is going to be the fact that this is going to cause recruitment of those cells that are needed to clean this area up. So again, those recruited cells are going to be mostly your phagocytes, come in and swallow up any pathogens or toxins. And they have this increased blood flow and leaky capillaries to help them get to the tissue. So there's, in addition to increased blood flow and leaky capillaries, we mentioned this term chemotaxis. And chemotaxis, again, is a chemical signal that cells will follow. So you have these cells floating through the blood, macrophages, and neutrophils, etc. They can crawl out of the capillary and they're going to end up making their way toward these inflammatory mediators through this process of chemotaxis. So chemotaxis increases, and these cells are drawn specifically toward the presence of things like cytokines and these inflammatory mediators. Another really interesting part of the recruitment of these cells is called margination. So margination refers to the fact that some of these inflammatory mediators are going to cause the walls of capillaries to become sticky. So as a cell is passing through a capillary, it's going to hit these sticky parts of the walls, and instead of just going right through that capillary and on out to the venous circulation, these cells are going to stick to these sticky capillary walls and make them less likely to be able to crawl out and toward the site of the infection. So chemotaxis and margination are two properties of cells during the inflammatory response that are going to help to get those cells out to where they need to be to assist in defeating an infection. So once these phagocytes are affected by the inflammatory mediators, that's going to take them through these steps of activation. So you have local macrophages in addition to those local mast cells. Mast cells, again, being kind of the cells that sound the alarm by sending out inflammatory mediators. There are also local macrophages, which, as their name implies, are phagocytes. And they're going to become activated during this immune response. The other, along with those local macrophages, the other first responders are going to be your neutrophils and monocytes. And they're going to travel toward the direction of an infection through chemotaxis. Neutrophils already being capable of efficient phagocytosis. And then monocytes are basically just an immature form of these macrophages. So when monocytes travel toward the site of infection, once they reach that area, they're going to mature into macrophages. And that includes some changes in the proteins that they synthesize, for example, that make them more efficient. So once they reach that site of infection, they kind of activate within themselves, this response that's going to make them more efficient phagocytes. And then in addition to those cells, there's going to be production of new white blood cells that's ramped up as a response. And this is going to occur in the bone marrow. So the last part about the inflammation response is that part of this injury or an infection can lead to a fever, which is defined as a body temperature over 99 degrees Fahrenheit. So this is actually going to serve a physiological purpose related to immunity. It's thought that an increase in body temperature can increase the efficiency of phagocytosis. So in essence, the kind of working theory is that phagocytes are better at doing their job of clearing infections at higher temperatures. So that's kind of thought of as being the most likely explanation for why we get fevers in the case of an infection. But how it comes about is that when you have an infection, either bacteria or the cells that are damaged themselves are releasing substances that cause an increase in the set point of your body's thermostat in the hypothalamus. So normally the hypothalamus keeps your body temperature through negative feedback homeostatic mechanisms at about 97, 98 degrees. And these pyrogens, these substances that are released by bacteria or damaged cells, increase the set point up to a higher temperature. So what this is going to do is it's going to cause the hypothalamus to recognize what would be a normal body temperature as being too low, and activating negative feedback in order to increase the temperature back to this new set point. So that's why you're going to have chills, for example, this contraction of your skeletal muscles that generates a lot of heat, and that heat is going to raise your body temperature. So the hypothalamus is regulating temperature homeostatically, and those pyrogens just increase the set point. So basically they turn up the temperature on the thermostat of the body. So in this last phase of the lecture, we're going to be talking about the adaptive immune system. And going back to what we've already talked to previously, the adaptive immune system allows your body to respond to the presence of certain antigens that are associated with a disease state or an infection. So in the adaptive immune system, you have the cell-mediated responses and you have the antibody-mediated responses. And beginning with the cell-mediated, the cells that are mediating this response are the T cells. And remember, those are going to be maturing in the thymus and found in the lymphoid organs. And there are two types of T cells. There are helper T cells, which activate other immune cells. So helper T cells, once they're activated, they're going to release cytokines. And these cytokines, again, are messengers that are going to stimulate the activity of other immune cells. So these T cells don't have a direct effect of killing pathogens, but they do stimulate the other parts of the immune system to mount a defense. And the other type of T cell are the cytotoxic T cells, which means they are toxic to other cells. They directly kill other cells. So stimulating immune response by activating other cells, and the cytotoxic cells are themselves the killer. And like all parts of the adaptive immune system, again, this activation of T cells is going to be based on the presence of an antigen. So here we have a T cell and a cell that has an antigen present. Now, this can be an antigen presenting cell, so like a dendritic cell is a phagocyte that's going to destroy a pathogen and then hold out a piece of it, the antigen, and present it to other cells like these T cells. So an antigen presenting cell can be a cell that interacts with a T cell and activates it. And that's a way of sounding an alarm, basically. The antigen presenting cell says, hey, look at this antigen. You need to find this and take care of it, because this is a piece of a virus or a bacterium. Aside from an antigen presenting cell, this cell that's activating T cells can be a diseased cell. So this can be a cell that's part of your own body. So like a cancer cell can be targeted for destruction by the immune system. And this disease cell is going to have antigens on its surface. And that can also target and cause activation of the cytotoxic T cells. So the T cells are going to contain a receptor called a T cell receptor that's going to recognize one specific type of antigen. Now your body is going to produce T cells with a variety of different T cell receptors. And in the case that a T cell is activated by binding to that antigen, that T cell is going to increase in number. It's going to replicate itself. so that your body can mount a response against that antigen. And after that, after exposure to this antigen, you'll have effector T cells, which are dealing with the problem at hand, and memory T cells. So of the T cells that have the capacity to bind to a unique antigen, only the ones that meet that antigen are going to exponentially increase in number. Because why make T cells that can bind antigens that you're not actually being exposed to? And these antigens can be from a foreign, like a bacteria that's been destroyed by something, like a phagocyte. So let's say a phagocyte has ingested a bacterium, and now it's presenting an antigen. Once it's presented that antigen, it can come in contact with T cells, and those T cells, again, that recognize that antigen will increase in number. So this allows our immune system to become well versed in dealing with the types of species of microbes that are likely to infect you, so that if you get infected again, you can now mount an efficient immune response. So that would be an antigen presenting cell, is going to digest whatever the invading pathogen is and present an antigen that's considered exogenous. So antigen presenting cells are going to display exogenous antigens, which mean they come from outside the body. Now, there are other antigens that are considered endogenous. So in the case that you have a diseased cell, like a cancer cell that you want to get rid of, the antigen that it's presenting is an endogenous antigen. It comes from inside your body. So in that way, you're actually going to be targeting the cells of your own body based on them having this antigen. That's your immune system has learned to associate within a diseased state. And one other component is you have the receptor on the T cell, the antigen, but the antigen is going to be bound to a protein in the plasma membrane called an MHC protein. And the only really important thing to note is that there's two types of MHC proteins. And for these cytotoxic T cells that are taking care of our diseased cells, they're going to bind MHC type 1. And the helper T cells are going to bind MHC type 2. Let's take a look at the mechanism of action for a T cell. Once a T cell is activated, meaning this T cell with its T cell receptor recognizes an antigen on some diseased cell or some foreign cell that it needs to clear. Also could be a cell that's infected with a virus or an intracellular pathogen. Sometimes bacteria can get inside the cells and infect cells as well. So this T cell has recognized this antigen and become activated. What it's going to do is release a compound called perforin. And what perforin is going to do is going to perforate the plasma membrane of this abnormal cell. So it's going to poke little holes in that cell. And it's going to then release digestive enzymes. So you get release of these enzymes by the cytotoxic T cell. And since there are holes in the cell membrane, those enzymes can travel inside this cell and basically eat it from the inside out and destroy that cell. So once this has been accomplished, this cell is dead, its antigen is gone. This T cell can then move on and seek out other cells to destroy. unlike cytotoxic T cells, the helper T cells, again, are just there to stimulate other cells of the immune system and ring the alarm. So when you have an activated macrophage, a phagocyte that's an antigen-presenting cell, it's going to present this antigen from some foreign invader. And it's going to present it to these helper T cells. So when a helper T cell is activated, what happens is, instead of killing this cell, which is actually an immune cell, it's going to release cytokines, which again are those chemical messengers of the immune system. And those cytokines are going to spread out around, and they're going to have a variety of different effects on the cells around them. Those cytokines that helper T cells produce are called interleukins because they have effects on other white blood cells. Inter meaning between, leukins meaning leukocytes, between other white blood cells. And they have a few different effects. They can affect cells of the innate immune system, or they can affect the adaptive immune response. So one of these interleukins, called interleukin 3 or IL-3, is going to stimulate macrophages. So this is a macrophage that's an APC. Let's say there's other macrophages out here that are phagocytes that are going around taking care of an infection. So the helper T cell stimulates them by increasing the rate of phagocytosis. So it's going to make these macrophages more likely or more efficient phagocytes. So just increasing their activity. And it's also going to have an effect that's a positive feedback mechanism. So these macrophages, once they've been stimulated, not only are they better at phagocytosis and more efficiently clear out an infection, but they release their own interleukin, which is going to in turn stimulate more helper T cells. More stimulation of more helper T cells, they're going to be releasing more interleukins, and this is going to feed back on itself until you have a clearance of the infection. The effects that helper T cells have on the innate immune system is going to target the cells of the innate immune system, including other T cells and B cells, which make antibodies. So helper T cells are going to have a direct effect on B cell proliferation. Now, what proliferation means is when a B cell proliferates, that means it's going to reproduce and create more B cells. So an activated helper T cell is going to cause B cells to increase in number directly. Now, it's also going to have effects on the the adaptive immune system through interleukins. One in particular, interleukin 2, is going to increase the number and activity of cytotoxic T cells. So those other T cells that are directly killing other cells are going to be proliferating and increasing their activity based on the presence of IL-2. And there are other interleukins that are also going to cause B cell proliferation in an indirect way. But these other interleukins can also cause B cells to ramp up their production of antibodies. So again, a lot of these mechanisms seem like positive feedback, and that makes sense in the case that you want to quickly get rid of an infection. Is to deal with it as quickly as possible, you want to stimulate as much immune activity as possible. However, a hyperactive immune system can also be a bad thing. So the last bit of the adaptive immune system includes the B cells. And these are particularly relevant. You probably heard a lot about whether or not COVID patients have produced antibodies that are thought to make them immune to future infections. So this is really important part of the immune system that plays into whether or not you'll be immune to viruses if you're exposed to them in the future. So B cells produce antibodies, and antibodies have a number of effects that help to provide immunity, and we'll get into those soon. So when a B cell is produced in the bone marrow, it's also going to mature in the bone marrow. So you can remember B stands for bone marrow, and that's in contrast to the T cells, which mature in the thymus. So B cells that generate antibodies that are self-reactive are going to be destroyed before they leave the bone marrow. So B cells eventually make it into lymphoid organs where they can participate in immunity. However, if you have B cells that are producing antibodies that bind to self-antigens, meaning antigens that are present on your own cells, that's going to target them for destruction, and that's where you see autoimmunity. So autoimmunity means that your immune system is targeting your own cells. And there are a variety of autoimmune disorders. So self-reactive antibodies can be a very bad thing that can cause disease. And so in the bone marrow, there's a mechanism in place to destroy self-reactive B cells, or B cells that produce self-reactive antibodies. So when a naive B cell, and we say it's naive if it has not encountered its specific antigen. This is kind of like T cells, where we mentioned that your body produces many different types of T cells that each can have the capacity to recognize a unique antigen. Same thing with B cells. The body is constantly producing B cells that can recognize a variety of types of antigens. But most of those, after a few days, if they don't encounter their antigen, they're just going to be, they're going to die off eventually. However, if a B cell is exposed to its antigen, remember the lymphatic system is sucking up these bacteria and immune complexes, and those antigens can make their way into a lymphoid organ where you have B cells. Once exposure to an antigen occurs, if a B cell is exposed to its unique antigen, it's going to differentiate into two types of cells. A plasma B cell, which is going to actively produce antibodies. And antibodies look like these little Y-shaped molecules. So it's going to actively produce antibodies that are going to circulate in the blood and provide immunity in the short term. But memory B cells are going to exist to provide long-term immunity. So, as I said, these B cells are kind of turned over constantly if they don't encounter their antigen. But if you do encounter an antigen, those memory B cells are going to replicate. You're going to get a lot of these memory B cells, and they can stick around for months or years or even a lifetime, so that when you encounter that antigen again, your immune system already has the B cells in place to produce antibodies. So these memory B cells can become activated and quickly ramp up production of antibodies that will target a specific antigen or pathogen. So it's really important that for establishing long-term immunity, that you can come up with memory B cells that produce antibodies that will be effective in targeting the pathogen that your body encounters. So let's now look at the effect of antibodies. And the first effects are going to be one that you probably have heard before in lab, which is agglutination. So remember from the blood lab, you apply antibodies that are going to attach themselves to an antigen that's present on a specific blood cell. So when they meet that antigen, they're going to cause those blood cells, or whatever those cells are, to agglutinate, to stick together. And in the case of an infection, that is very preventative. It means that the cells that are kind of stuck to one another can't move around freely, so that's going to limit the infection. But it's also going to increase the chance that they're found by phagocytes. So agglutination is going to lead to easier phagocytosis and also prevent those bacteria or whatever the pathogen might be from spreading as easily. Another effect, so let's say that the antigen is a small free-floating molecule, or something like a virus, which are very small. So if antibodies, which tend to clump together, are bound to their antigen, they are going to pull those out of solution, meaning they're going to cause everything to clump up and form an immune complex. And these immune complexes are much easier to clear via phagocytosis than if these antigens were just floating around on their own. So it's going to target these antigens, whether it's a toxin or whether it's a virus. It's going to cause them to be targets for phagocytosis. So both of these are going to cause elimination of whatever that antigen is or whatever it's bound to, if that happens to be a pathogenic cell. Some other interesting effects of antibodies are in the case that you're talking again about something small, there can be neutralization of whatever that antigen is. So going back to this little cluster of antibodies bound to toxins, let's say this toxin has a particular active site. So part of that toxin, part of the molecule itself is what causes toxicity. These antibodies being bound to that toxin can make it less toxic. They can neutralize the toxin. And let's consider the example of something like the coronavirus. So if we say this is the novel coronavirus, and you've probably seen all the pictures with the coronavirus and its spike proteins on the outside. Those spike proteins allow it to enter cells and cause infection. So by blocking those spike proteins, if antibodies recognize that antigen, if the antigen in this case is the spike proteins, antibodies can stick to the antigen and effectively block whatever purpose it has, which in this case is entering cells. So if you have a virus, a coronavirus, that has antibodies stuck to its spike proteins, it's going to be much less effective at infecting the cells of your body. So this is one reason why it could be so important to have these antibodies present, and how that could confer immunity. One specific way that antibodies can increase phagocytosis is by directly interacting with both the antigen, say on the surface of a pathogen, and also interacting with the phagocyte that's doing the phagocytosis. And this process is called opsonization, which just refers to making it easier to eat. So in this opsonization process, this antibodies are also called immunoglobulins because they have kind of a globular protein structure. But so immunoglobulins or antibodies are often designated IG and then a letter. In this case, this specific type of antibody, IgG or immunoglobulin G, is going to help with opsonization by binding both the pathogen and the phagocyte, bringing them close together and promoting phagocytosis. So opsonization is just going to increase phagocytosis. Lastly, antibodies can also activate the innate immune system. So again, the innate and the adaptive immune system don't exist in isolation. They can cooperate with one another. And this is the case when certain antibodies can either activate the complement system, which again, the ultimate goal of the complement system is going to be to activate complement proteins and in turn activate the membrane attack complex that's going to end up killing pathogens. So antibodies can activate that complement system, and they can also stimulate the production of inflammatory cytokines and other inflammatory mediators. Those mediators being released by mast cells and basophils. So certain antibody, IgE, we talked about IgG, this is immunoglobulin E, can bind to mast cells and basophils and cause them to increase their production of inflammatory mediators. So these are all the effects of the adaptive immune system, which is remarkably efficient at dealing with specific pathogens and gets stronger with repeated exposure. So this is why vaccines work, is because you're exposing yourself deliberately to an antigen that you expect to be present on a pathogen. So that'll do it for our overview of immunity. And lastly, I want to talk about a couple of different types of immunity. So antibody conferred immunity can be either active, meaning that you are yourself producing antibodies, but there are important circumstances or situations where it's actually necessary to receive or beneficial to receive antibodies via passive mechanisms, meaning you're not producing those antibodies yourself. You're receiving them either from your mother, first, let's say. So a newborn infant gets breast milk from mom. Well, that breast milk is going to contain beneficial antibodies that help that newborn to fight off infections. That newborn's immune system will be fully capable eventually. But early on, the cells of the newborn's immune system are going to be naive to many pathogens. And so that's why young people are quicker to get sick, is because they haven't been exposed to those antigens that are part of these pathogens. So they need to build up immunity over time. And to help them when they're really young, they get antibodies through the breast milk from mom. Another type of passive immunity that I'm very hopeful can be a coronavirus or COVID-19 treatment is delivering antibodies intravenously. So you may have heard that one idea for the treatment of COVID patients is that you can have patients who have recovered from COVID-19 who now create their own antibodies that help them to fight off the coronavirus. Well, those antibodies are present in the plasma. So if we harvest plasma from recovered patients and introduce that into a patient that's suffering that hasn't built up their own immune response, that might, by introducing those antibodies passively, we might be able to confer resistance to that infection. So passive immunity, I'm hopeful. There haven't been a lot of great results as far as using treatments. The idea right now is that we won't beat the coronavirus until there's a vaccine potentially. But I'm pretty hopeful that delivery of antibodies from recovered patients can help to passively provide resistance to coronavirus. And those are in clinical trials right now, so we'll have to wait and see.